Welcome to section 14.2, where we try to figure out this question of where did life come from and how did it early on proceed to allow us to get to the point that we have complex multicellular life like us today. Now, to look at this question, let's first go back and talk about the discussion that they had trying to figure out where life came from. This was back in the 1700s, 1800s. And at that time, the popular theory was this idea of spontaneous generation. And this was that in the right conditions, life would be created from inanimate objects. So in other words, I could put rotting meat out or broth out, which themselves are not alive. But in those conditions, it would allow us to get flies and maggots created in the case of rotting meat. It would allow us to get bacteria created in the case of broth. And this was evidenced by the fact that if they put rotting meat out, they shortly would see maggots and then flies. If they left the broth out, they would shortly see bacteria growing in it. Now, the competing theory was biogenesis, which is that life makes life, that ultimately it's the flies that land on the rotting meat, that lay the eggs, that grow the maggots, that grow the flies, or that it's bacteria being carried by the air or on our bodies that gets into the broth that then multiply. And so now the broth becomes overrun with bacteria. Now, when they did the studies, and this is kind of cool about what science was, if you look back in the 16, 17, 1800s, you had where Francesco Redi was one of the first scientists, and he took an open container and he put rotting meat in it and showed that we got maggots and we got flies. He then took and covered it with a lid that was essentially like a cheesecloth that prevented the flies from getting to the meat. And he was able to show that as long as he prevented the flies from getting to that rotting meat, there were no maggots, there were no flies. So this seems to point to the fact that flies are what make flies, not rotting meat. Otherwise, we should have had maggots and flies in the container covered with cheesecloth. Now, some people said because you're blocking stuff, you're messing with stuff, because there were later experiments where they would go through and boil broth, so that way it was killed all the bacteria. They would then seal it and show that it wouldn't go bad. But people said this active sealing stuff was messing with this vital essence or vital force that created life. And so Pasteur is the one that ultimately sealed that biogenesis was the winner, where he would take and create these elaborate swan neck flasks. And so the flasks were never sealed, but because if you've got air and bacteria coming in, they're just going to collect in this bottom part. They can't make their way up this thin, narrow neck. So he was able to make it where he'd boil this broth, use this elaborate neck, and then the broth would not go bad because none of the bacteria in the air could get up the neck of this flask. He could also show that if he broke the flask, that ultimately would go bad very quickly because now the bacteria are able to essentially fall in from the air, and so the bacteria could then grow in the broth. And so this kind of sealed the win for biogenesis, but it created a problem. If life comes from life, where did the original life come from? With spontaneous generation, it was convenient that non-living things could make living things. So we didn't have to worry about that question. It was built into the answer. Well, now that we know that's not the answer, we have to figure out where the heck did life come from. And ultimately, what that comes down to is a separate theory from evolution called abiogenesis. In reality, it's more of a hypothesis at this point because we just don't have as much evidence to say exactly what happened, but we certainly have a lot of evidence that shows many different ways it could have happened, and so that's usually considered to be a fairly good evidence that it likely would have happened. So abiogenesis is us getting life from non-living material, and most people peg this down to having occurred due to something called the primordial soup. And this primordial soup is effectively that in these early Earth conditions, if we go back, 4 billion years, that in those conditions we would have had organic monomers present. And this can be explained by the fact that we find organic monomers on material that arrived from asteroids and meteors. This can be explained by the fact that organic monomers would have formed naturally on Earth back then. But regardless, this primordial soup idea is that organic monomers would have been present, and in the right conditions in the early Earth, that they would have formed organic polymers, which allow these organic polymers to stick together like we see with the liposomes, where many of them, just to be energetically stable, like to form certain structures. You'll see liposomes, or lipids in general, like to form these spherical bilayer structures, which we could recognize as effectively being a cell membrane. I mean, it's very, very, very much the same idea. And so we then get these cell-like structures as these polymers kind of interact together. And if these cell-like structures, like these liposomes, happen to have the right things inside of them, if they happen to kind of capture the right guys inside when they form, we could now get a very primitive early cell. So these cell-like structures can do many of the characteristics of life. They can grow as they add more lipids. They can break apart into separate things. You get reproduction. Some of them, depending on what's inside, we have pieces of RNA and protein that can act as a metabolism. So these cell-like structures can already do many of the things that life does. But to do everything life does, that's when we get our first cell. Now, in a lab, because this occurred over the span of 600 million years, maybe more, because we peg life around probably 3.8 to 4 billion years that it occurred. So if the Earth is 4.6 billion years old, we need oceans containing lots of these organic monomers, polymers, these cell-like structures, and we need them to be kind of sitting around for 6 to 800 million years for us to really run this experiment again and make sure that that gives us a cell at some point in time. So this is incredibly difficult to do. That's why I don't know if we'll ever move this out of the realm of hypothesis and fully put it into the idea of theory, where we can be almost certain of exactly what happened. What we do instead is we try to disprove things, to say, let's just show this couldn't happen. And what we find is for many different things, we can't disprove it, which means if there's many different things that are possible, possibly even probable, we start to get a pretty good idea that this would have happened, that this will happen elsewhere in the universe, even on other planets, other moons perhaps, that life may not be as rare as we think, that in the right conditions, life could actually be almost close to a certainty if you have the right conditions for a long enough amount of time. It's kind of like buying enough lottery tickets. If you buy billions of lottery tickets and billions of lottery tickets, at some point it becomes likely that you're going to win, even though individually the chance of winning a single lottery ticket is small. But if you have enough of these different cell-like structures, organic polymers around, and you've got a long enough time, it can start to become almost a certainty.